Um, Thanks everyone for tuning in to this month's CTR seminar series. Today we will be hearing from the Sentinel research team on a project that's very relevant to the current environment. And without further ado, I'll pass it off to Dr. Rennard.
Okay, thank you, Jared. If we could take the first slide, if you could go ahead and start the video. This video shows a time lapse of the spread of SARS-CoV-2 that leads to the disease that we call COVID-19. And as you can see, it started in China, but then it spread to Europe, United States, Southeast Asia. And by February, it was recognized. It took a few months before people really believed it was important. Mitigation efforts started in a variety of places, and you can see that within another month, in fact, it reached pretty much the whole world. And in some places, like the United States, the disease had become really rampant. Other areas where the disease had increased rapidly, such as in Spain and other parts of Europe, seems to slow down. Other places like Brazil that got a late start on the pandemic increased rapidly. And you can see that 6 months after the start, in fact, there's millions of cases in the United States alone. And it's really a major global health problem. Of course, it's increasing exponentially and up to the current time. I think the video stops a little bit before just now, hopefully here in just a moment or two. Okay, so this is what we've experienced. Now, the problem, go to the next slide. The problem that we're trying to address is that epidemics spread exponentially. And this is mostly non-intuitive. At the beginning, it's hard to recognize that it really exists. And of course, people formulate opinions about that, which can carry over. The rate at which it spreads, of course, being exponential, it spreads faster than most people are going to expect. And then COVID was particularly difficult because it was spread by asymptomatic persons, and it has a long incubation time, which means that it was a perfect storm for rapid spread of a disease. Next slide, please, Jared. So what we're going to do today is I've just defined the problem, and we're going to present our approach to a problem of how would you get an early warning system for a problem like the COVID pandemic. That is, how would you know sooner that it really existed and know sooner where outbreaks are occurring? I'll outline the approach and then lead to and introduce then the other members of the team, all of whom are required to deliver on an important goal like this. They'll each present their portions, the portion of which where their expertise is particularly important in this project. And then I'll conclude with a current overview of the project status. So Jared, go to the next slide, please. So if you take a look at Nebraska and you take a look at SARS-CoV, the traditional way to diagnose an epidemic is you have to recognize cases. And they have to have something that's striking so that you can recognize them, either a pattern of illness or their severity or their numbers. And if you have only a few cases, and you can see where I've drawn the little S's, which means symptomatic cases in Nebraska, then it might be hard to recognize when there's only a few. And this is particularly a problem for something like COVID, which is a novel pathogen where you don't even really know how to recognize it. So the problem is not only that we want to have an early warning system, but we need an early warning system for something that we've never seen before. Now, the next slide summarizes the approach. In contrast to the map I drew before, our approach takes advantage of a concept called homeokinesis. And Dr. Yentis will introduce this in some detail. But the general concept is that healthy systems are resilient. That is, they have stable adaptability. And transitions from this stable adaptability, either by becoming too rigid or becoming unstable, represent a transition to disease. So our approach was to develop an inexpensive wearable sensor that could detect this transition, for example, through the loss of the complexity and the information in the signals that a sensor could detect, to detect transitions from health to disease. What this would provide for COVID, if you go to the next slide, is that you'd be able to detect all of the asymptomatic cases, which means that there'd be larger numbers. This could, in fact, tell you that an outbreak is occurring, and that outbreak is occurring earlier than you might otherwise have expected. Even a 2-week or 4-week head start on mitigation efforts for COVID could have had a huge impact. It could also help resolve the issue of when mitigation efforts could be relaxed. Next slide. So what's needed to deliver on our hypothesis, which is that a wearable sensor-based system can detect physiologic indicators that will diagnose a transition from health to disease with SARS-CoV-2 infection. So what's needed is a sensor that can detect the relevant signals, a way to extract meaningful physiologic parameters from those data. We then have to validate those parameters as diagnostics. And so to do that, we need a clinical study for data collection and rigorous statistical analysis plans that allow those data to be interpreted. And so this then leads to our team. Go to the next slide, Jared. So Dr. Eric Markvicka is an assistant professor in the Department of Engineering at UNL, whose expertise is the integration of microelectronics into durable textiles, which is just what's needed to have the kind of wearable sensor which is required. Dr. Jenna Yentis is an associate professor at UNO, and her expertise is in the extraction of high-order physiologic parameters using a variety of techniques, in particular nonlinear analysis from time sequence sensor data. We then need to validate those parameters, and Dr. James McClay is the local principal investigator for our PCORNet site and an expert in database management. And he's exactly the right kind of person to be able to organize the kinds of clinical studies that will be required for validating this approach. And finally, Dr. Ying Zhang is the chair of the biostatistics department in the College of Public Health. He's a statistical methodologist, and in fact, the statistical requirements of this pose some very interesting challenges, which are important scientific questions in their own right. So this introduces our team, and with that, if we go to the next slide, Jared, I'd like to turn it over to Eric, who will present the first portion of the talk. Eric?
Great, thank you, Dr. Rennard. So yeah, again, my name is Eric Marvichka. I'm an assistant professor in the Department of Mechanical Engineering at UNL. My lab, the Smart Materials and Robotics Laboratory, is interested in creating new types of soft electronic materials to enable traditional electronics to intimately interact with the human body. Go to the next slide. So there's been major advancements in terms of the miniaturization of electrical components all the way from the mainframe era to the emerging wearable platforms of today that have brought computing power closer to the human body than ever before. This has unlocked a wide variety of novel applications such as wearable health monitoring. However, one thing that hasn't changed over the decades from the mainframe era to the emerging wearable platforms of today are the materials that are used to construct these devices. These devices that are closer to the human body than ever before are still primarily composed of metals and hard plastics. This choice of materials ultimately limits the placement on the human body to locations of low movement and flexibility as represented by the black pads shown here on the human model. And next slide, please. Recently, there's been major advancements in terms of the materials and mechanics that have enabled the creation of devices that mimic the properties of natural human tissue, but retain or are capable of the high functionality of these traditionally rigid devices. These devices boast properties such as thin, they're lightweight, they're soft, highly deformable and elastic. However, these devices require tethers that ultimately provide power and data communication in these devices. Next slide. Furthermore, their thin, ultra-thin, low modulus construction is inherently fragile. This generally results in single use of these devices. It's very similar, I feel, to trying to reuse saran wrap. It's very difficult and takes an immense amount of practice, right? I think my grandmother is the only person I know that can reuse saran wrap, and that's not very practical for sort of general population deployment of a wearable system. Next slide, please. Our work has been focused on creating new types of stretchable hybrid electronic architectures to enable the embedding of advanced microelectronic components with high performance textiles. These systems boast a lot of the similar properties as the previous slide, having thin, lightweight construction, structural conformability with the human body, but integrate the necessary components to provide untethered use. The integration with high-performance high-performance textiles enables a mechanically robust platform and allows the device to be reused. By interfacing the skin adhesive directly with the textile component, we can replace the skin adhesive after use. And then we can keep a lot of the sort of more expensive components, the electronics of the device. Next slide, please. Using this fabrication approach, we've demonstrated a wide variety of potential applications, from forehead temperature monitoring, a necklace that can monitor sounds of the neck for detecting food consumption, to human motion tracking, where this requires structural conformity, but also a stretchable platform when the electronics are placed across the joints that allows movement. Next slide, please. So as part of this team, we're working on creating the wearable Sentinel device, where we're integrating 2 sensors into a wearable platform, biopotential and accelerometer. This allows us to measure then electrocardiogram, respiration, and gait. Dr. Jenna Jens will then kind of talk about how we process these measures to understand these transitions from health to disease. With this platform, we're making advancements in terms of the fabrication to create a sealed, waterproof construction to enable deployment in a general population for sort of a multiple month observational human study. Next slide, please. My introduction to the team was through Dr. Jens, and we met at the Science and Engineering and Medicine Retreat in March of 2019. This was 3 months after I joined the department. This was by far one of the most productive meetings that I've attended so far. And the team has then brought me provided a lot to me. For me personally, I think some of the most interesting research is happening at the intersection of engineering and medicine. So the team has provided clinical relevance. They provided access to key populations and a wide range of expertise, from medicine to disease, expertise in clinical validation of medical devices, observational human trials, and really cutting edge physiology, signal processing, and data analysis. Next slide, please.
So I think I'm up next. I'm Dr. Jenny Yentis. I'm an associate professor in biomechanics at the University of Nebraska, Omaha. I've been there for 6 or 7 years now. So yep, you can go on to the next slide. So my portion is really trying to make sense of the data that we get out of these sensors. So the topic or the theory that I like to use is homeokinesis. This is a theory introduced by MDs, and so it helps it be a bit more clinically relevant. It's the ability to maintain some sort of ordered system that fluctuates within a range. So a lot of people are familiar with homeostasis, which is trying to keep a certain number. So you're looking for a mean value and you're trying to stay around that value. Homeokinesis is more interested in the fluctuations that are happening within a range. So here we just have heart rate plotted on the y-axis within a range of 60 to 80 for some type of resting heart rate. And you can tell that through the 3 plots that I've given, the fluctuations within them are quite different from having no fluctuations, just being a mean value of 70, to having quite a few severe fluctuations from minimum to maximum to those that sort of fluctuate within. All of these have about the same mean, but really the point is the fluctuations really are critically important. And if you could go on to the next slide. So ways that we can look at the fluctuations, one of them is we can look at the actual variability of them. People are really familiar with this, in standard deviations or coefficient of variation just gives you a magnitude of how much the fluctuations are moving. So like I said, the means were very close in these, and you can see that the standard deviation of the middle and the left one are fairly similar. And then on the right, you have no standard deviation because there's no flexibility there. So this might not provide enough clinical information, just having information about the variability. That just might not provide enough because you can tell that the middle one and the left one are quite different from each other, but their values that you're getting out are quite the same. So if you go on to the next slide, another way that we can look at the fluctuations is the complexity, or sort of what is the information that you're gaining within the fluctuation. So if you think of something that's perfectly repeatable, such as the one you see on the right, it gives you no information. If you're familiar with the concept of entropy, it's kind of giving you information about the order and disorder within the system. So you're really gaining no bits of information when it's always the same. And then if you look at the far left, it might be a bit more random, so you're gaining a lot more information. So really the idea of complexity is this idea of how much information, probability you have within the data. And another MD, Lou Lipsitch, who's at Harvard, proposed a theory called loss of complexity. He originally proposed it in the idea of aging. So as you age, you lose this complexity in many biorhythms. However, we've noted that that happens a lot in pathology as well. So a good idea to look at is, are you losing this complexity by becoming too predictable or by becoming too random? Because randomness really isn't complex. So there's sort of this nice optimal level there that you're aiming to get that gives you some information about health. And if you can go on to the next slide. And another piece of information that we can use with these signals is coupling with biorhythms. So this is something that I work on quite a bit, and I look at walking and breathing rhythms and how those are coupled to each other. But I provided several topics or papers that have been published that really focus on these coupling, and like the one at the top, uncoupling of these biological oscillators may be indicative of a type of syndrome. They give information about metastability. They give information about other different conditions. So it's not just walking and breathing that can be coupled, but any type of biorhythms. They're all communicating with each other, but how do we quantify that? And how do we quantify the information that's being shared across these rhythms? So my work is really focused on trying to extract clinically meaningful data from these signals. So if you go on to the next slide. So really what we will do is we'll take the data that's gained from the sensors that Dr. Martin-Vicka is going to put together. And then I gave 2 examples of, at the top there, those are step times walking, and the bottom, these are interval times for breathing, I believe, in seconds, yes. And so that is an idea of how do we look at the variability and complexity of each of these signals independently, but then how do we look at the coupling between those signals? And we also will have electrocardiogram data as well, so then we can look at coupling potentially between the different combinations of 2 or 3 signals at a time. Let me go on to the next slide. So they asked us to talk about the team and how we met. I met Dr. Rennard about 12, 13 years ago now when I was a PhD student and I was interested in COPD, and went and sat in his office and listened to him give me all kinds of information that I was fascinated with. And we've been working together basically ever since. And so he's really provided me a way in which I can look at things from a clinical standpoint. There's times that I've talked about things, he's like, well, that won't work in medicine. And so it's really nice to have a clinician that will give you those, that perspective. And then combining all of medicine, engineering, data analysis, access to patients, having statistics, that gives you the capacity to actually complete these really nice projects. And the encouragement and motivation that you get from the team is quite nice as well, because you get quite excited when you're on a call talking and everyone is starting to generate ideas. So I think that that's a real benefit too, as well as having a team like this. And I pass it off to the next.
Well, that would be me. Hi, I'm Jim McClay. I'm a professor in the Department of Emergency Medicine. 30 years practice of emergency medicine, but my focus has always been on biomedical informatics. So this is the obligatory disclosure slide of what I'm doing. And I'm just going to start out right now because this lecture is in the context of our IDEA Clinical Translational Research Network, where we're really kind of focused on developing team science in the translational area. So it's very exciting to see this group gel and to bring something to fruition that fulfills one of my problems in emergency medicine, which is to answer the question, is this person sick or not? And so I first met Dr. Rennard a few years back when I was looking to get a clinical investigator for a pulmonary medicine project that is part of a research platform that we've been building that I'm going to describe, that gives us connections to clinical communities and their data. So you can go to the next slide. Like I say, part of our clinical translational research, where I was director of the biomedical informatics component, is to support team science with varying ways of reducing the barriers for the team. And in clinical and translational research, that barrier is frequently access to clinical data and to patients. So we teach the bench to bedside to policy kind of translational science arc, and then our group has been building data infrastructure, putting in place the policies and procedures and skill sets to make this work. And so if you go to the next slide, the project that I've been directing is the clinical research analytics environment at UNMC, which is a set of tools and processes to participate in large-scale clinical research and translational research projects. So it creates this lovely, well, I call it lovely because it's my baby, collaborative platform that we have put together, and we participate in national research networks, including the Patient-Centered Research Network, the National COVID Cohort Collaborative, and the National Center for Advanced Translational Sciences, the Accrual to Trials Network. And this is, in each of these cases, in order to make the clinical data available, we not only have to go through the process of extracting the data and linking it to various data sources, but also if you're looking at clinical data, it's a special class, and there are many more policies and procedures around the protection of clinical data than other types of data. So not only do you have all of the big data problems, but you now have a lot of security and policy issues to deal with. So by working through those processes in advance, it really reduces the barrier to spinning up a study like this. So we have an environment that where we put together the processes and policies to support translational research, and a multidisciplinary team of informatics specialists and programmers who know how to operate on clinical data, but also we have a technical platform that contains the data that we've extracted from our clinical partner, Nebraska Medicine. But it is also rendered in exactly the same format as all of our other participating sites around the country. And then we build education and training on top of that. So if you go to the next slide, this particular opportunity came up. Dr. Rennard and I have been in discussions on opportunities for some time, but when the idea of actually implementing and studying a tool like this, we can look through our portfolio of activities and say, well, there's a study going on right now, which is looking at high-risk healthy people who are exposed to the COVID virus and are likely to convert to being infected. And this is the healthcare worker exposure response and outcome trial that is funded through our patient-centered research network. And we already had, because we're participating, we already had the various data use agreements and IRB agreements in place. And so we were able to go directly to the study team and suggest this Sentinel trial as an ancillary study and get approval and support in a couple of days. So that reduced the barrier to let our talented engineers and analytics and statistical people study that without having to wait for the months it usually takes to get these permissions in place. So that's what I was bringing to the team. So you can go to the next slide. I think I'm done with my presentation. You are muted, Dr. King.
I'm meeting from 7 o'clock until now.
Yes.
My name is Ying Zhang. I'm the currently professor and the chair of the biostat department. Been here about a little over a year. So next slides. I basically want to talk about the role of the biostatistician in kind of team science research. Things like we're gonna, as a group, our member gonna involve more and more this study. I want to let people kind of let people know. I mean the actual role we can play above beyond just the sample size calculation power. Okay, just conventional stuff. So I mean, first of all, be a research about statistical research. I mean, you definitely need to have a scientific mindset. So I mean, I'm so grateful to Dr. Rennard. He actually, he and I start to have conversation only like 2 or 3 months after I joined the UNMC. So came to my office. I was impressed by his, by his enthusiasm in the research, all kind of great ideas. I learned a lot from him. So I enjoy talking to him for not like a particular topic. Generally, I gather a lot of clinical knowledge from him. So I mean, it's clearly, as I said, it's very important. It's a genuine interest as a biostatistician in the scientific research. Okay, so and also a lot of time when he talked about, he brought me a lot of stuff. I haven't heard about it as a statistician. So I mean, it's very interesting, and I mean, I can imagine it can be very impactful. The stuff, I mean, he brought me. So basically, another important feature to be, to have as a statistician is a willingness to be challenged. Sometimes you don't know it, but you're willing to learn and you want to catch up and you want to exercise your strengths to it. And also, it's very important, it's tied with, if you want to be challenged, you also have a curiosity and a desire to learn things from the other. So that's why I learned a lot of stuff. So this thing, obviously, I have no idea, previously what the guys do. So I learned from Eric, from from Jenna, what they're doing. I mean, things like, very intriguing. It's kind of prompt my interest into this particular research. Obviously, I mean, it's most important. You have a drive to have a scientific discovery. So as a statistician, certainly we cannot play the solo game to have a good impact for scientific discovery. So you have to be in part of the team in part of the member in the team science. So it's a very, 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 very beneficial to multiple parties. Next slide, please. So how we gonna actually, this is I'm talking about the general procedure, the engagement in team science. In team science, it's not like a team science. You just have a buddy from the statistics in our team. That's the team science. No, it's not. We need to be engaged in the time science as early as possible. So I so appreciate, in the beginnings, Steve and I talk about multiple times the idea, and also we start with the multiple projects, right? So we narrow down to that particular one. It seems like it's more promising based on the availability of the data and also the manpower. So then clearly it's going to be, first of all, identify to stimulate. This kind of early team discussion is going to stimulate the scientific premise for the projects, right? And also for us as a statistician, from this scientific premise, how are we going to derive the testable hypothesis? You can develop the hypothesis, obviously, transition the question to the hypothesis. Whether you can address this hypothesis, that's another level of the effort. Okay. Then obviously, once you have the hypothesis, we need a brainstorming the technical approach. This is also very important. It's not trivial problem. It's like a two sample comparisons, things like that, right? It's not a typical randomized clinical trial thing. So it's a very challenging. I mean, I certainly as a biostatistician, I like to be challenged. I mean, can kind of stimulate, my interests. I mean, after we have I think at least a month of discussion, then look at the data. Jenna sending me the data, so I kind of look at the data. So try to reshape this. I mean, the hypothesis and the specific aims. Then what? Then you have to develop the research plan, right? Develop the research plan. Need to identify the primary outcomes. Jenna gave me a list of the outcome. I try to find a most sensible outcome that can address the question. Okay. Then obviously, because of this is about the early detection of somebody might be sick based on physiology data. It's not so trivial, right? So I did quite a bit of little research. I do have a brief knowledge to tackle this kind of problem, but I have to confess, I don't have an expertise in that particular area, but I know, how people doing with kind of multiple physiology time series data to try to make a reasonable detection of something may going wrong down the road. Okay, so that's exactly fit the thing of these projects. Then I kind of, after read quite a bit of literature, then I come up with a detailed technical approach plan. So next slide, please. So here we go. So for that particular project, so I did a little bit of preliminary analysis to try to identify the most sensible physiology outcome from basically it's a variability complexity. So then I talked with the team, mainly just Dr. Rennard have the primary analysis plan and also the backup analysis plan if that doesn't work. So basically, it's the supervised learning method or unsupervised learning method. You don't have the relatively kind of outcome. So you're kind of monitoring the data, the multivariate array of the data, physiology data, continuously try to, at any point, detect a possible changing point, right? Changing point indicates something going on, right? So technically, I mean, what people do is kind of how you define the changing point detection. You have to come up with the methodology. So statistical methodology, I come up with kind of very popular one called the likelihood ratio methods. Kind of bunch of data in the beginning and in the prior this changing point to be detected changing point. Then the bunch of data after to be detected changing point. It's kind of monitoring the threshold. You know, the data is gonna, you know, the bigger the ratio indicating more likely this time is possible is the changing point, right? So, I mean, that's just the idea. So how we're gonna compute that statistic is gonna be the cumulative sum, the Q-sum algorithm. That's very, very useful in the quality control, you know, to find something go bad, okay? So, but that's just the technical part. So, you know, you don't really know, you know, whether the stuff you develop is justifiable, it's right. So you have to have a validation stage. So I kind of developed an LC analysis procedure for the validation. So having said that, because this particular population is a healthcare worker, so if initially we thought about COVID-19, right? Because as you may say, for that population, I mean, the incidence of COVID-19 infection, it's very low. So, I mean, this is the caveat. So this kind of methodology, it's not going to be a magic to give you very accurate answer, right? Maybe you're not getting much going on because the low incidence. So we have to have a backup plan. So now, I mean, Steve and I basically talk about, we have a different plan, so we call it a secondary plan. So we're basically just collecting all respiratory infection, not necessarily COVID-19, can be anything, right? So then you may increase the, I mean, increased events that will help, you know, to detect the meaningful changing point. But how do you know? I mean, this changing point relates to the COVID-19. Then when we have the data with the event, then we can develop another type of like learning methods, unsupervised, called the latent clustering method. I mean, to single out the trajectory specifically tailored for COVID-19. But I mean, that's just a plan. Whether it's going to be successful or not, we'll wait to see. So that's pretty much I just want to talk about. So I have to say, you know, kind of the scientific interest, like Dr. Rennard presented to me, is very contagious. All right, so it's his passion to me. So allow me to be involved with him of all kind of research projects. If I'm not available, I will identify my people to work. I think our group, since I'm the chair in the department, I think we need to develop the powerhouse for this type of biostatistician to be able to engage in a team science in a significant role. It's not just like a service person. Okay, that's all I need to say. Thank you.
Okay, so what I'm going to do next is I'm going to give some comments about, from my perspective, of how the team came together, and then reflect on, then update people on sort of where the project is at the present time, and then we'll have time for questions. I think from the previous, previous four presentations, that we're really in completely different domains. You can see that there's absolutely essential things to deliver on a complex and important project like this that are absolutely required in order to deliver the goal. So how the project came together from my perspective is that Jenna's mentor, Nick Sturgio, suggested that she come speak to me about her PhD topic. And that led to me being on her thesis committee. And as Jenna commented then, we've really had a collaboration since then. Her research is focused on the coupling of respiration and gait in general and in COPD specifically. And frankly, that's been a wonderful opportunity for me. I've learned a ton of stuff from Jenna. Hopefully it goes in both directions. And one of the things that I think that everybody mentioned is during their individual presentations is how the team, people in the team learn from each other. Everybody on the team has substantial specialized expertise that other people on the team do not know. The other people on the team, though, are very eager to learn from each other, and that's worked very well. Now, as part of the collaboration with Jenna, we had developed a device which frankly was extraordinarily kludgy that could be used to capture data for the kinds of studies that Jenna wanted to do in COPD. That device was patented. UNMC has actually licensed it out, and it's keeping Jenna fairly busy. But Jenna understood the limitations of her device. She then met Eric, and she described how that happened at a university device. It's kind of a mixer trying to bring scientists together. They learned of their mutual interests, and that part of the collaboration started. Now, Jenna and Eric didn't mention that they've submitted several grants, one of which that Eric led has just been funded. There's several that are unfunded, but that collaboration, which was designed to develop more sophisticated sensors to answer Jenna's questions, was in flight. Now, I've heard Jim present at infrastructure sharing meetings on multiple occasions. For people that have never tried to manage large data sets, it's really impossible to imagine the complexity involved in doing that. If the data set extends into multiple institutions where the data management systems differ, the complexities rapidly become mind-boggling. That kind of infrastructure is absolutely necessary for any kind of a big project. Jim is really a master at has been a master at getting UNMC involved in that. And as he mentioned, he's managed to engage me in several projects, not just me, but he's been extraordinarily effective for the institution as a whole, and clearly has a key role in this specific project. And then Ying was recommended to me by one of the members in his department with whom I have a very cordial and productive relationship as potentially a very good person to engage in this project. And I'll say that Ying was perfectly willing to accept a cold call from me and invite me to come to his office. This was in the pre-COVID times, so that we could chat about the research project. He was willing to engage completely, to listen to what the question was, to then offer suggestions, and then, as he described, to work together towards bringing the team to a reasonable project. So what it offers, specifically for me, is that this is novel science, and it's novel science aimed at what I think everybody recognizes an important problem. In addition, and this is also really important, is that there's absolutely delightful collaborators. The collaboration to date has been fun. Now, it'll be much more fun if we get the money, and it'll be even more fun if we succeed in what we plan to do. But just the process of going through has been okay. Now, you do want to get the other things too, and I'll summarize where we are. And so if this is going to happen, there have to be reasonable grant funding opportunities. One of the natures of academics in general is that you have to find your own money, and so it's not reasonable to work on projects where you can't find it. And that's a bit of a challenge for the Sentinel project. Next slide. So where we are currently is that I think there's two fundamental problems. And the first is the somebody else's problem. For people that are Douglas Adams fans, you'll recognize the quote, but the way to make your spaceship invisible in the Hitchhiker's Guide to the Galaxy is you surround it with a somebody else's problem field, because nobody can see somebody else's problem. Now, that's part of the problem with getting funding for a project like Sentinel that involves all of these different domains, because the engineering is a key domain. But frequently the engineering, people reviewing engineering grants don't understand about all of the other things that are required. And you could say the same thing for physiology or clinical or even statistics. As Ying pointed out, a lot of people think statistics is just looking up sample sizes. In fact, there's really important unanswered questions about statistics. And clinical means not only understanding a disease, but also understanding how we deal with the information relevant to that disease in a real world setting. And so people generally in one domain have difficulty seeing other people's domains, which makes getting grant funding difficult. And then there's the bacon and eggs problem, which is maybe not quite as famous. It's maybe not kosher, but where the bacon and eggs problem is that if we only had eggs, we could have bacon and eggs if we only had bacon. So of course we could do Sentinel if we only had all 6 of these things. And so that's usually a bad idea in an experiment, since you can get support for one thing, but we need support for a lot in order to do it. So the fundamental problem with Sentinel, next slide, which is no different than any other academic pursuit. Have we gone to the next slide that's got the dollar signs? I can't quite see that. Yes, we have. Is where do we get money? Okay, so next slide, please. So we have a number of opportunities, and it's always important to sort out where the opportunities are. They don't all need to be pursued, but if there are no opportunities, it's probably not a good idea to work on it too hard. So the University of Nebraska has collaboration grants, and this is the grant that Eric was successful at applying for that will fund the development of a device. Now, it won't be exactly the same as the Sentinel device, but it'll be close enough that will dramatically advance what we're doing. Now, we have submitted Sentinel to the Department of Defense. We also would like to send in to NIH cohort ancillary studies where patients have been assembled, the data have been put into a data-friendly thing, which Jim thinks is at least a little piece of nirvana. It makes it easy to get access to relevant data that have not only been cleaned, but have met the whole variety of regulatory requirements in order to do this kind of research. And we're planning to send in an NIH director's application for emergency transformative grants relevant for COVID. But each of these grants can fund different things. And we need to have a bunch of different pieces to make the whole thing work. We need a device, we need the physiologic parameter extraction algorithms to be working. We need the clinical studies piece and the management of the clinical information, and we need the appropriate epidemiological and statistical approaches. And if you take a look at the various grant sources, the Sentinel project will really focus on the device and the clinical, but it really won't talk about the implementation of this, that is the epidemiology. And the physiology that we're presenting, as Jenna presented, is really off the shelf. It's not pushing the physiologic agenda. In contrast, the NIH Director's Award could push the physiology. It might actually be able to do everything. On the other hand, an NIH cohort study, you really can't even begin to do it unless you already have the device. Now, NIH will fund physiology and it might fund clinical questions, but it's not going to fund big epidemiologic questions. The Sentinel device will detect something bad is happening. It could be anything. It could be COVID, as Ying pointed out. Our approach, we also should be able to detect influenza or air pollution events, which makes it really an ideal epidemiologic study, which EPA, for example, or the Health Effects Institute would be delighted to fund. However, they will not fund the device development or the physiologic development, although they might fund the last 2 bits. So the question then is, how do you put together a funding program that can, in the long run, support the whole package? And what's needed for that, I think, is a few things. First of all, you need to have delightful collaborators, because you have to be patient and you have to be persistent and you have to try more than once. But we're in process and we're enthusiastic. And I think that if you go to the next slide, I think that the next slide should be the invitation for people to ask questions, not just of me, but for anybody else on the team. Thank you.
Great, thank you, everyone. Do we have any questions? We have one in chat. Oh.
Yeah, I will say, if you can still hear me, that that I am will be very depressed if nobody asks any questions.
Dr. Sharani has a question in chat. She writes, given the urgency of this pandemic, I was wondering how the sensors that were pointed out might be fundamentally different than the already widely available mobile apps for measuring O2 saturation, heart rate, gait parameters, etc.
Okay, so let Eric answer that, and then Jenna as well, because the sensors differ, but also the kind of data that are collected differ, which means that you can learn more stuff. But I'll let Eric and Jenna address that.
Yeah, so thank you for the question. So we're looking at measuring electrocardiogram and also triaxial accelerometer data at the chest. And so there are smartphone platforms available for measuring this, such as the most advanced Apple iWatches. However, they're measuring electrocardiogram at the wrist, which is really not the best location to monitor electrocardiogram. We should be measuring this at the chest. This can result in sort of additional noise and potentially faulty data that could lead to misdiagnosis and unneeded treatments that could result in additional stress for these patients. So we're creating devices that allow access to locations on the body that are not possible. And that allows us to integrate some of these clinically grade electrocardiogram sensors to monitor this activity.
Sorry, I was waiting for a dog to finish barking. In addition to the sensors being in different locations, the data that we're collecting from the sensors is slightly different. So some of the data, like let's say for gait parameters, they're giving you maybe the number of steps or that type of thing. We're very interested in continuous data. So more or less, we call it gait, but it's more or less movement. We're using accelerometers to get this continuous type kind of data so that we can look at the coupling continuously. The same with electrocardiograms. We're interested in having the full signal rather than just heart rate, beats per minute. Even though my example was beats per minute, that's not really the best for us to gain that information. A lot of heart rate variability is done between the intervals between the RR peaks. And that is really what's important, is that variability between the RR peaks really, it has been shown over and over again to determine mortality and other issues. And in addition to that, the respiratory data that we're getting is we're actually pulling that from the EKG. So we are using, some of the sensors come with that built in. So we're looking at, if we don't have a sensor that commercially pulls those data out, how to do that mathematically. And that's been shown over and over again in papers as well. We're focused on those three to start with, but we can add other sensors for other things. And to find the best combination that gives us the most comprehensive information. And that's really sort of the end goal, whether it's for COVID or another potentially diagnostic or predictive platform.
Great, thank you. We have a couple more questions from chat. I'll just answer them or ask them in the order they were sent in. Dr. Fruehling asks how have you adjusted to working as a team, going from face-to-face meetings to Zoom and distributed work environments, and if there's any team science methods that help this. And Dr. Rennard, you seem to have kind of brought this together, so maybe you want to take the first stab at it.
I don't think we did anything special. You talk to each other and you try to listen to each other. You send a lot of emails. I think we did try to divide up the work and so forth. So it wouldn't have been any different than the way we'd have come together from any other project. Obviously, some of us, Jim in particular, but Ying as well, have tremendous experience working on team projects. So, this wasn't the first rodeo for some members of the team. And so, as you can tell, there's a varying mix of people at different maturity in their career levels. And that's probably a good thing. But I don't know that we tried to follow any specific team procedures other than trying to get the grants done on time.
I would quickly add that I think we met we did a lot of Zoom calls. There was weeks where some of us were meeting 3 times a week as we were trying to put out a grant fairly quickly. When the DoD call came out, the turnaround was quite fast. So we were meeting quite often, and I think that really helped, whether it's in person or not, our communication, and sometimes our email strings, I get lost in them. So we were really trying hard to keep our communication as going as possible. But of course, there's always things that could be improved.
Great, thank you. And we had a question about any data privacy issues that arose and how they were addressed.
The data privacy issue, when it comes to clinical data, is foremost in our considerations, and we have numerous mechanisms to avoid exposing any identifiable information about our patients or our participants in studies. So if it's a study participant, they will be consented to share their data with us. And of course, then data privacy is just the typical keeping things encrypted and secured. When we're looking at large populations, we have to remove all patient identifiers and substitute in something else. And that is, it's been a great, Steve or Dr. Rennard mentioned I'm on other teams, and one of the biggest teams is this national effort to create a linked set of electronic health record data in which we have to go through these privacy preserving practices in order to make the data available for researchers to access it without excessive barriers. And so the data is known as de-identified data, and as far as the IRB is concerned and HIPAA, it is not an issue to analyze that data.
And I'd be curious on that same note, from an informatics perspective, if there's any location sensitive data in the device technology, or is that restricted out? Like, this isn't smartphone reliant or anything like that with the device?
Yeah, so the data will be transmitted to a smartphone device in near real time and uploaded to secure cloud-based storage.
Okay.
We also transmit only the bare minimum of data. So there's no patient information that gets transmitted. It's usually just the sensor ID along with the data. So it's a string of numbers that come through in some sort of file that's not even labeled. So if you were to open it, you would get a string of numbers and you wouldn't know which column is heart rate or which column is this. It's tried to transmit in the most general, well, vague way, I guess you should say, that limits your ability to go back and try to figure out what it was even, what that file even was.
Great. And then we had another question. Is this targeted more toward mild or severe COVID cases?
Yes, so I'll take that one. It's not really targeted for mild or severe. It's targeted to look at a transition from health to disease. So people with severe disease, it should detect. We very much want it to also detect asymptomatic infections. How we will do that, of course, this is where healthcare workers who are being monitored very, very closely provide a really important clinical tool. It also raises some interesting statistical questions. That is, how do you detect the change from somebody? So somebody might subsequently become symptomatic, but maybe the physiologic parameters will be detectable 3 or 4 days before. This is what Ying was addressing when he was talking about some of the more sophisticated statistical approaches, such as change point analysis. So the idea is that it would be able to detect mild and severe COVID cases, which is part of the challenge.
Well, the point is, for the severe cases, obviously, better way to detect, right? Exactly. Regular, you want to basically screening. It's more like a screening tool.
Yeah. We don't want to miss those cases.
Yeah.
But the value really comes from detecting the milder cases. Yes, that's absolutely correct.
All right, we are at the hour. Any final questions before we wrap up? If you do have any, please send them to me and I will triage with Dr. Rennard.
Thanks, everyone.
Yeah.
All right, thank you.
Thank you. Thank you for having us.